Hello, everyone. My name is Kaylee Marcano, and welcome to my final microbiology project. Bacteria are often perceived as tiny and simple, yet, their remarkable diversity, complex behaviors, and adaptive capabilities reveal a level of sophistication that is fundamental to microbiology. In this presentation, we will systematically examine this characteristic and unravel the process of identifying an unknown bacterial species. So, let's begin. The objective of this assignment was to use observational data, selective and differential media, biochemical tests, and microscopic analysis to determine the identity of an unknown sample. At the start of the project, the unknown organism could have been one of the several coliform or non coliform bacteria. To guide the identification process, I used a dichotomous key, essentially a flowchart of yes or no questions that narrow down possibilities. Without the dichotomous key, interpreting multiple test results could become chaotic. But using it gave a structure, clarity, and a logical pathway toward the correct species. The next step was to gather experimental evidence through a series of carefully selected laboratory tests to reveal specific characteristics of the unknown organism. One of the first and most important steps was the gram stain. This technique separates bacteria into gram positive or gram negative, depending of the structure of their cell walls. Under the microscope, my sample appeared as purple, round-shaped, coxy arranged in clusters, which is consistent with gram-positive Staphylococcus. There was no motility observed, and the morphology strongly suggested the genus Staphylococcus early on. Next, I tested the organism on EMB and McConkey agar, both selective and differential media designed primarily for gram-negative enteric bacteria. My organism showed very slight growth on both media, but no significant color change. This suggested two important interpretations. One, the organism is not truly gram negative because it did not thrive. And two, it may have some ability to tolerate the inhibitory components of this media, something occasionally seen in gram-positive cocci like Staphylococcus species. The lactose fermentation test delivered an unexpected positive result. Typically, a positive lactose test points towards coliforms. However, there are documented cases where certain Staphylococcus epidermidis strains show limited or weak carbohydrate fermentation. The mannitol broth test came back negative. This was a highly significant result because Staphylococcus aureus ferments mannitol strongly, while Staphylococcus epidermidis typically does not ferment mannitol. On MSA, my organism grew well, expected for Staphylococcus, which tolerate high salt concentrations. The medium showed that initially looked like a positive reaction, but when compared to the mannitol broth results, it became clear that the slight color shift was not due to mannitol fermentation. The coagulase test was negative, which is one of the most definitive steps in distinguishing between staphylococcal species. This is due that Staphylococcus aureus is coagulase positive, while Staphylococcus epidermidis will be negative. This final biochemical confirmation aligned perfectly with all previous results. Now that we have found out what is our unknown bacterium, what is exactly Staphylococcus epidermidis? Well, is a gram-positive coagulase negative staphylococcus. It's found primarily on human skin, where it forms part of the normal microbiota. It thrives in salty, dry environments like the epidermis, and it is usually non pathogenic, but can be opportunistic, particularly in hospital settings. It is known for forming biofilms on medical devices, such as catheters and prosthetic joints. In conclusion, this project demonstrated how microbiological identification is both an art and a science. Even when individual results seem surprising or ambiguous, each test contributes to a piece of a larger photo. My final poster compiles the full identification process, including microscopic images, media results, and biochemical test interpretation. It serves as a visual summary of how each test contributed to identifying the organism with confidence and scientific accuracy. Finally, I want to give a special thank you to my professor, Dr. Juan Morata, to the lab staff, and all the organizations that made this project possible. Their support, guidance, and dedication made this an incredible hands on learning experience. And thank you for watching.